Okay, so I'll get us started off. Welcome everyone to the first meeting of our colloquium this semester. I'm Aaron Clausette. I'm an associate professor in the department. It's my great pleasure to introduce our speaker today, Orit Peleg. She is an assistant professor in computer science and also faculty in the Biofrontiers Institute. She received her bachelor's in physics and computer science and her master's in physics, summa cum laude, from Bar Ilan University in Israel. She then completed her PhD in 2012 at ETH Zurich with a thesis entitled Simple Models of Competitive Interactions in Biophysical Systems. She went on to do a postdoc at Harvard University until 2017, and she joined us here at Colorado in 2018. Her research focuses on collective dynamics, where we seek to understand how group and population level behaviors emerge from the interactive dynamics of individuals. These ideas have broad implications for our understanding of complex systems of all kinds, including human society, economies, and even ecologies. And today, Orit will tell us about her recent work in a talk titled Physical Computation in Insect Swarms.
You're on mute.
Sorry, I just stopped sharing. Okay, can I just get a confirmation you can see my screen and can hear me? Okay, great. Thank you so much, Aaron, for the lovely introduction. And I'm really excited to have the opportunity to tell you what me and my lab members have been up to over the last 3 years. So the topic is going to be physical computation and communication in insect swarms. But before I dive in, I want to give you a slightly, take a step back and give you a slightly broader view of the problems and the questions that drive research in my lab. As humans, it's easy to forget how difficult it is to communicate using just natural resources. We already got used to having mobile connectivity and we can pretty much reach anybody at any distance. We also have a bunch of backup systems like postal mail, satellites, landlines, and so on. But animals actually have to continuously solve similar problems using only natural resources that they have in their local environment. And of course, many animals evolved very beautiful and elegant way to communicate with each other. There's tactile communication, visual, chemical, vocal, excuse me. And of course, it's going to be relatively short range in comparison to the distance between the pairs of organisms that are interacting. But animals also evolved really elegant solutions to communicate over longer distances in larger groups. And there's many beautiful examples of this behavior. And I'm only going to name 2 examples just to give you a taste of it, which I'm sure you actually already know. So the top example, the movie here on the top right, is a movie of slime mold. So these are social amoeba, and what you see here, all these white dots are actually food sources. Now that I'm playing the movie, the social amoeba are actually using chemical communications to guide other members of their society to the food sources. And actually, they also solve the problem of the shortest path between these food sources. So in a sense, that's a bit of a physical computation in that sense. Another famous example is these beautiful flocks of starlings. So very large number of birds that have been shown to, just by using communication with their neighboring birds, are able to make quick decisions about turns, and which allows them to effectively move in these really large groups. So I think there's still a lot to learn from natural systems that had eons of evolution to perfect some of these tasks. But on the other hand, I also like to keep in mind that these systems serve as inspiration for some technologies. For example, swarm robotics, where also individual building blocks, individual robots that are minimal and affordable communicate locally to achieve some global function. We also have, of course, a few members of our faculty working on these kind of systems here in our department. Okay, so my goal is to understand how communication signals in nature are generated and interpreted, And I tend to keep in mind concepts from physics and information theory like detectability. So how to maximize signal to noise ratios, or compression, minimizing energy and minimizing bits to represent information, robustness, so how sensitive the whole system is to failure of individual nodes. And my approach, well, I'll just reveal it. My approach is to use computational modeling and mathematical modeling, very specifically theories that can be actually tested with behavioral experiments with the model organisms that we use in my lab. And as you can see, we're using insect swarms, fireflies, and honeybees, not just because they're, at least in my opinion, incredibly cute, but because they're really pushing the limit of communication in large groups, so thousands and tens of thousands of individuals. And also, just by looking at these animals, we can say something about what they're communicating as, and that will become more clear as I go through some specific examples. Okay, so I don't have time to talk about everything we've been doing over the last 2 years, but I chose a couple of examples that I think would highlight the gist. So I'm gonna tell you about some chemical communication in honeybee swarms, specifically how honeybees use pheromones to locate their queen, and how in the context of chemical communication, they're able to exchange food with each other and distribute resources in a distributed way. And I'm going to switch to visual communication, where I'm going to use Fireflies as a model system. Here the questions will be more about signal design and how to increase signal to noise ratio, either by synchronizing the message or designing the signal or the message in a way that is distinguishable. Okay, so let's dive in to honeybees. For those of you who are not familiar with honeybee swarms, they're very important pollinators, also for humans' sake, and they live in very, very large colonies of tens of thousands of individuals. There's one queen, there's a couple of hundred drones, and then the rest are female workers. So obviously, because there's only one queen, the queen is really important for the survival of the entire colony. And they really operate as super organisms in the way that they are making decisions. For example, how they're using the waggle dance to communicate information about food sources and nest locations. Also more physicsy kind of communication. For example, they can sense locally the temperature at the entrance of the hive and ventilate their hives according to that information. They can create these really large congested aggregations that are part of the reproductive cycle, which are things that I actually worked on during my postdoctoral studies. But all of these are already assuming that the colony is coherent. There's a bunch of bees that are aggregated around the queen. And the questions that we wanted to pose is actually, how do they become a coherent colony, a coherent swarm in the first place? How do they find the queen and aggregate around her, given the fact that they're actually using pheromones to locate her, which decay very rapidly in time and space? So it turns out that the bees are using chemical communication also to locate the queen, but they're actually amplifying the signal that is coming from the queen and creating their own pheromones. So can you see my cursor? Yeah, great. So I'm placing now my cursor on the abdomen of the bee. This is where their nasenov gland is, with the pheromones that they use to locate the queen. They have antennae that they use to sense those pheromones. And then they use a really cool trick, which is to fan their wings on top of these pheromone glands. So they're actually drawing air on top of this gland and moving these pheromones backward in a very specific direction that depends on their body orientation. And from our preliminary experiments that were led by Dumai, who is an graduate student in my lab, we noticed that bees not just go all the way to the queen, which would maybe benefit themselves, but they actually stop at a certain distance, and then they start this behavior, scenting. So they start producing the pheromones and fanning their wings. And then other bees would come from further away and start this behavior. So there's this propagating amplification of the signal. But these were kind of anecdotal, and we really wanted to be able to test this behavior and understand it more quantitatively. And for that, we worked together with Greg Stephens from OIC in Japan, and also Michael, who is another graduate student in our department. And the idea was really to create a high throughput classification code that would allow us to automatically detect the bees that are scenting. And this way, even if we work with really large swarms of thousands of bees, we can very quickly say which one is scenting and which one is not. So the training set kind of looked like this. We had short videos. Here on the left is an example of a bee that is scenting and producing pheromones. And on the right, there is a bee that is just kind of hanging out, grooming. And I don't have time to go into all the details, but we used neural network classifier that at the end, actually, the output was the position of the bee that is scenting, whether it's scenting or not. And also, it gives us the body orientation, which is really helpful for studying this behavior. So armed with this high throughput classification, we can, I'm going to show you how a typical experiment looked like. So the queen is here in the top right corner in this cage. The rest of the worker bees are placed here at the other corner. This is a semi-two-dimensional system, so we're looking at it from the top. This is aerial view right now. And the experiment is simply placing the queen and the bees in these locations and seeing how the bees interact with the queen and aggregate around her. So as you can see here, the bees, this is a bit sped up. You can see that the bees are kind of exploring. There's a lot of scenting. So all of these blue arrows are bees that are producing these pheromones. So there's a sharp increase in the number of bees that are scenting. And then if I scroll through a little bit faster, then at the end of the experiment, most of the bees actually aggregate around the queen. Okay, so this was just one observation, but actually wanted to quantify this a little bit more carefully. And specifically, we wanted to ask, are the positions and locations in which the bees are scenting and producing these pheromones are really indicative to where the queen is? Do they really provide directions for other worker bees in how to reach the queen? And the way we tested this was by taking all of the scenting events. Here I'm just illustrating, so all of these arrows are scenting events in this illustration, and I'm going to treat them as gradients, as local gradients of a hypothetical surface, attractive surface, where high values in this surface are should be correlated with where the queen is, if really all of these positions are leading us to the queen. And to make a long story short, indeed, when we take all of these local events of scenting in our experiment, the global map that the bees create, high values in this surface really correspond to where the queen is, which is great. So it means that the scenting really is somehow helping the bees to find their way to the queen, which is further away from where they were originally placed. And okay, so we know that there's a correlation there. And now the question is, what actually triggers the scenting behavior? How do the bees decide when to stop and amplify and propagate that information? And to answer that question, we look at a few, again, quantitative classifications of the behavior of the bees. One of them is showing us that there is a probabilistic decision-making approach where some percentage of bees are going to become scenting bees. Rather, for example, another scenario where maybe there's a fixed number of bees in the colony, no matter how many bees we have in the entire experiment. So this is kind of what you're seeing here. So the y-axis is the number of bees that are scenting. The x-axis is time. The color, the level of green correspond to the number of bees that we had in our experimental arena. And you can see that the number of scenting bees is going to be proportional to the number of bees that we have in the arena. So that's one. The second thing is that we noticed that the distance between the bees that are scenting seems to be pretty much consistent around 6 centimeters, no matter how many bees we had in the arena. So that tells us that if a bee, basically to interpret this, if a bee started to scent, then there is a particular probability in terms of the distance as to where another bee might be stopping to scent. And since we're dealing with pheromones that diffuse over time, then a particular distance would also correspond to a particular pheromone concentration. So it suggests that perhaps the bees are being triggered by some threshold of concentration above which the bees decide to scent. Okay, so now we're going to take these two experimental observations and use them to build an agent-based model that would help us to understand better this entire process. And the model goes like this. So we have, again, like in the experiments, a two-dimensional arena. Our agents move randomly until they encounter a pixel where the concentration of the pheromones exceeds a certain threshold. And then here comes the probabilistic aspect. So 50% of the time, they're going to perform a directed walk, where they're going to go up the pheromone gradient. And the other 50% of the time, they're going to produce their own pheromones and amplify the signal. And to do that, we just use a classic solution to the diffusion problem where there is a directed bias. I'll be happy to explain this more if people are interested in the Q&A, but maybe more visually to see this is how an agent that is producing pheromones in an isotropic way, so in the same amount in every direction. And this is, and this is how it looks like for a directed pheromone signaling. Okay, so we put this all together. We ran a bunch of simulations, which was actually really helpful in terms of determining the robustness of this entire aggregation process. Specifically, we were changing some behavioral parameters that the bees could presumably change by themselves in their behavior, like the threshold concentration that triggers their scenting, or the directional bias that they create with the fanning of the wings. And we see a couple of pitfalls in a sense that if, for example, the threshold concentration is too high, then of course no bee is going to scent in the simulations and no clustering is going to occur around the queen. If it's too low, then actually the bees tend to get stuck in these local equilibria of small clusters that are really hard to get out of. At the intermediate values, we're observing a diffusion limited aggregation, which would scale really badly as the size of the system increases. And then it turns out that there is a sweet spot here of a combination of these two behavioral values where we can see clustering via percolation. So here's an example of that simulation. The bees are scenting. You can see that the signal was propagating all through the system. And then using these set of parameters, they're actually able to achieve more efficient aggregation around the queen. But it's only available when we have directional bias. So it's really highlighting why directional bias is useful here, and it helps the robustness of the system. Okay, then so everything I just showed you was actually in a very clean sort of artificial scenario where there's a two-dimensional arena that has no obstacles, but of course in real life, either inside their hives or outside in nature, the bees are going to encounter unknown and unconstructed environments. And so we extended that work to also consider what the scenario where we have obstacles in the experimental arena. And the bees are actually pretty good in solving this problem. When there is one obstacle here, here's just one line. We also explored the situation where there is a maze. And then just a kind of a teaser for work that was done by another graduate student in Melab Golnar this summer. We can see that the bees not only find the way to the queen in complex environments, but they're also able to find the shortest path. So this is a classical Y maze experiment where the bees enter it and they can either go left or right, and they choose the right, the left lane, which is the shortest path to the queen. Okay, so that was one of the examples that I wanted to show you today. I think it's a good example to how behavioral experiments are helping us to design these kind of models, agent-based models, that helps us better understand the process. In this case, it's localization and aggregation around the target, the queen. And it also shows us that actually having the directional bias is beneficial for the bees, because we're seeing that in the phase diagram that I've shown a couple of slides ago. Okay, so moving from locating a target, I want to discuss another problem that the bees have to solve, which is distributing resources of food. And in particular, bees in a hive have many different tasks. They have some tasks inside the hive, cleaning, grooming the queen and the brood, or outside, guarding, and so on. And all of these tasks cost energy. So bees that are busy doing these tasks don't have time to actually forage for food sources or consume other sources of energy. And I just want to note as kind of as a side note that also, of course, that problem also exists in swarm robotics, where a known problem is that sometimes you just have to close the entire operation to charge the robots. Of course, people are working on it, but I'm mentioning it as a reference because I think it would be interesting to think about how the bees solve it, and perhaps adapt some of these strategies. So the way the bees solve it is by having a separate caste of bees that are foragers. So these bees are flying outside of the nest, and they collect nectar in their social stomachs, and then they come back to the nest, so they basically regurgitate some of these food sources. Sorry if this is a little bit gross, but I'm even going to show you a movie of how this looks like. So here we have two bees that are performing food exchange. It's called trophallaxis in the biological jargon. On the left, we have a bee that has more food in her stomach, so it's a fed donor bee, and it's sharing resources with this more deprived bee. Okay, so in terms of this process, there's an interesting challenge that a bee that just fed somebody has to solve, which is who to feed next. So if she chooses to feed a bee that is nearby, that would be one option, or if she can actually travel a little bit and feed another bee that is further away. And the reason this is an important dilemma is because if we think about the food distribution in the nest as a diffusion process of the food, we know from exactly just a very simple normal diffusion that the mean square displacement as a function of time is going to be a linear function of time, where the slope of this curve is going to be the diffusion coefficient. So if a bee is basically deciding to not move very far and very fast, it's going to have a lower diffusion coefficient, and it can have a higher diffusion coefficient if it actually moves further away. And the more you move, the higher the diffusion coefficient, the more mixing you have. So in that sense, our initial prediction was that we expect bees to move more and create more mixing in the system to more optimally share the food resources. So we wanted to understand this process, and we started with a fairly simple and naive agent-based model. This was, of course, I want to mention, led by Gollner, who is a graduate student in my lab, and also in collaboration with Liz Bradley, who is a collaborator and also co-mentor of Gollner. And the model goes like this. So we have an agent, again, performing a random walk, but now this random walk is actually going to be correlated, and we can modulate the effective diffusion coefficient of that agent by changing the turning angle distribution of the angles between consecutive steps. So when it's a very narrow distribution, this agent is going to move pretty much in straight lines. So it's going to move further away, higher effective diffusion coefficient, and mixing. and vice versa if it's a very wide distribution. Then when bees encounter each other in the simulation arena, if they're situated below a certain distance threshold, then they stop and exchange food, and they basically split the food that they have between them. So they come out of this interaction with equal levels of food in their stomachs. And we repeat these local rules of behavior until our distribution of food is uniformly is equal around between all the bees in the simulation. Okay, so here, this is really not quite a big surprise. This is really just following the logic that I told you about in the previous slide. So if you look at the convergence time, how long does it take for the bees to distribute uniformly the food within everybody in the simulation? So this is plotted as a function of the straightness of the random walk. So high values here would correspond to low diffusion coefficient. And we can see that indeed the convergent time is higher and vice versa when it's a higher diffusion coefficient. Just as a visual illustration of that, on the left side, we have a simulation with high amount of mixing compared to low amount of mixing on the right side. And you can even see by eye that the simulation with high amount of mixing reaches equilibrium faster. Okay, so that's our naive model. That was our initial guess. But since we have bees in the lab, we can actually test whether that's what the bees are doing. So these were experiments that Golnar ran over the last couple of summers. We have a combination of two groups of bees. Some of them are fed, and we mark them with pink dots so that we can also trace them throughout the experiment. And some of them didn't have access to food, so they're deprived. And we set the ratio between them to be a very small amount of fed bees, so that the bees in our experiment actually really face a difficult and urgent problem to distribute the food between everybody in the experiment. So again, it's a two-dimensional arena. We're recording it from the top. Here is a typical experiment where we just introduced the bees that are fed. And it's a little bit hard to see by eye what's going on, but this is where computer vision became really helpful. And we saw a couple of things that surprised us a little bit. The first was when we look at the trajectories of the fed bees, we see that in the beginning, they move quite a lot. So they're shown here as red lines. But as the experiment progresses, they actually stop moving. So they look more like dots. So they move less, their speed is lower, which is actually effectively lowering the diffusivity of these bees. Very counterintuitive to what we thought. And then another interesting thing that we noticed is that the bees actually cluster. So over here on this bottom line, you can see a connected component analysis. So we are basically plotting a few of these large clusters that the bees create. And more quantitatively here, I'm showing a plot of the cluster size on the y-axis as a function of time. And before we introduce any fed bees, we have very small clusters, but then as we introduce them, these clusters are becoming larger and larger. So we're not really sure what's going on, but now since we have the model, we can actually program in aggregation into the model and try to understand what's the benefit, if there is a benefit for aggregation. So this is what we did. We added one more rule to the behavior of the bees, where if bees are close to each other, then a certain threshold R, then they start moving towards each other. And the rest of the simulation was exactly the same. So I'm not going to repeat this. But changing the simulation was actually quite interesting. So now when we plot the convergence time as a function of the attraction range between the bees, we see that there's actually a sweet spot with a very particular attraction range where the convergence time is optimal, so it's minimal. And when we compare the cluster size distribution and time series in the experiments, as well as in the modeling, then we can actually evaluate or guess what is the R value that we have in the experiments, and it turns out that it's really close to that minimum. So at least in that sense, the bees are kind of poised close to the optimal position in this model. Okay, so I showed you another example where we combined mathematical modeling and experimental data with the bees. And I think this is a really good example of how we go sometimes back and forth between the model and experiments. More precisely, I think this was an interesting exercise because we showed that a more tightly constrained random walk at higher densities, including the attraction, so the attraction that leads to aggregation, can actually be beneficial for data distribution in this kind of multi-agent systems. All right. So this is what I wanted to tell you about chemical communication in honeybees. And moving forward, I want to talk about visual communication in fireflies. And we're going to focus on signal design and synchronization. So fireflies have a lantern in their abdomen. Here is a movie showing them flashing. This is a behavior that they do as part of their mating events. It's visual, it's approximately digital, so light on or light off. And it's also potentially traceable in large groups, as I will show in a couple of slides. So it's a really simple signal. The males advertise themselves with a particular pattern that is associated with their species. And then the females have to be able to perceive that species specific pattern, and then they answer back if they perceive it. So it's a really, it seems like a really great, simple system to start asking questions about detectability and signal to noise ratio or compression of the information and energy. And it seems like the majority of fireflies address these two challenges in two different approaches. One is to synchronize their flashes. So this is the movie that you're seeing here on the left. This is a bunch of fireflies that are all blinking at the same time. And another way to think about detectability without synchronization is to design a particular signal that even in the presence of other fireflies is going to be detectable, and then still five males from one species can advertise themselves even in the presence of other species. Okay, so I'm going to start with the synchronized fireflies. And as I'm sure many of you know, synchronization is quite abundant in nature, both for inanimate systems like these pendulum clocks, but also for living creatures. So we as humans actually synchronize quite a lot. In some parts of the world, people synchronize their claps at the end of concerts. Musicians also synchronize, of course. And fireflies are one of these typical landmark examples people usually talk about in textbooks as an example of synchronization in nature. So in these particular models, I want to just highlight a few here. This is the Kuromoto model, which is a classical model of synchronization. The main assumption is that individual agents are performing some periodic function, even when they're just by themselves, like the pendulum clock or the people clap. So it's a periodic behavior. And there's a particular period, and then there is a coupling between the phases of different individuals when they're put together in a large system. So here in this example, the coupling constant can be many different things. It can be a constant, so it's really just like a spring potential. It can be a function of the distance between the two individuals, or a more complicated function of the distance, and more and more and more. Actually, there's hundreds of extensions and modifications to these kind of models, but very little experimental data to validate whether they're actually working for fireflies. So this was the gap that we were hoping to fill. a project led by Raphael Safati, who's a postdoc in my lab, together with Julie. We worked with a species of fireflies at the Smoky Mountains that flashes in bursts. So they flash a few times and then quiet for a while and then flash again. Here is an example of recording from our field trip. So what you saw now was a burst. So there were a lot of flashes. Now it's the quiet part. And here the fireflies are going to start another burst. And there's quite a lot we can already learn just from one view and looking at the temporal data. For example, if you plot the number of flashes as a function of time, we see two very distinct behavior when we compare low firefly density and high firefly density. So at low fireflies, there's a few sprinkles of flashes here and there. There isn't an obvious pattern, but as soon as there's enough fireflies, then they synchronize and we can start seeing this burst pattern. So we can quantify that, for example, by looking at the variance of the number of flashes in each one of these plots, which would have a very different scaling law when it's a random process, for example, a Poisson distribution, in comparison to something that is more like a clustering distribution. But as we saw before, in these mathematical models, the distance between fireflies can be really important. Which is why we went through the effort of recording the fireflies in stereo vision. So we have two cameras, either positioned more classically outside the swarm. This is what's shown here on the left side. Or we also have a setup where we place cameras inside the swarm, but there are cameras that have a 360 field of view, so they can actually kind of see the swarm from the perspective of an individual firefly that is in the swarm. So there was some additional math that we had to develop here, but it works pretty well. And as a side note, I want to mention that because these are relatively affordable cameras, these are GoPros off the shelf, and they're easy to use, then we also have trying to create a crowdsourcing project together with funding from National Geographic, where it's going to help us both acquire data in a really cool way, but also can really help firefly conservation. Okay, so we have stereo vision. We can reconstruct the flashes of fireflies in 3D. Here's one example of this. This is actually the first ever 3D reconstruction of firefly flashes that is shown here. And if I just scroll through, this is all the data that we got in the first round of experiments, which is really helpful because now we can look, for example, at spatio-temporal correlations, which is what I'm showing you here. So the y-axis is the distance between flashes, the x-axis is the time. And we can get these distribution functions and start thinking about how to plug them back in to these mathematical models. But there's actually also longer range spatial correlations that are really beautiful to watch. And I'm going to show you, the one way to reproduce this kind of signal relays that we saw in the field. So when we have longer range recordings of about 30 meters, we saw that the onset of the burst has a wave propagation, a spatial wave propagation. So it's a little bit hard to see in this movie, but I'll play this again. The start is here on the left and it propagates to the right. And then if you look at the average of all of these points, this is how it looks like on average. So there's really clear propagation of onset of flashes that moves at least 10-folds faster than the typical speed of flight of an individual firefly. So it's not created because fireflies are moving, it's created because fireflies are actually communicating with each other. And for some reason that information is moving in a very particular direction in space. Okay, so this was some information that we learned about the collective fireflies behavior outside in natural environments. But if we want to understand how fireflies interact with each other, then it would be helpful to know what individual fireflies are doing in more controlled environments. which are these tents that are shown here at the top right, that we brought with us to the field. And we had a similar stereo camera set up in those tents. And now we can actually track individual fireflies and start asking some questions about how they interact with each other. And one of the things we looked at was how do they align their bursts, because that's one of the very obvious synchronization behavior that we're seeing outside. But the surprising thing was when we looked at the interburst interval, which is presumably the period of each individual firefly. And as a reminder, the models actually assume that when we take individuals from the swarm, they would still have a very precise and characteristic interval. We see that that's actually not the case with fireflies. So if I'll just go in the reverse order here, when you have 20 fireflies, they have a pretty consistent peak around 10, which is 10 seconds, which is about the times which we're seeing outside also in natural habitats. But as the number of fireflies decreases, if we have 5 fireflies, then this distribution becomes wider with a longer tail. And individual fireflies don't seem to have any consistent interburst interval, and it's almost seems like they're just randomly flashing and kind of sampling their environment and only converging on a persistent interburst interval when they get a response from a sufficient amount of fireflies. So my colleagues and I are currently working on adjusting some of these existing agent-based models to account for that behavior as well. So for this particular example, currently we acquire behavioral experiments and analyze that data and found a few aspects of firefly behavior that we're trying to take into account now in refined mathematical models of synchronization, like the short and long range spatial correlations, and the fact that individuals don't really act as oscillators when they're isolated from the swarm. All right, so we talked about how fireflies synchronize as means to increase the visibility of the males. And finally, as the last example of projects in my lab, I want to talk about signal designs for fireflies who are actually not synchronous. There's quite a lot of data in the literature about firefly flashes. Here is an example from a paper from 2015. We know what their typical flash pattern is, and we also know how they evolved. So over here on the left side, this is a phylogenetic tree that is based on the genetics of these fireflies. And we're starting to ask questions about how did these signals evolve given the ecological constraints that we know exist, because different species must be distinguishable for the male and female mating behavior. So this is a project led by Chantal, who is a postdoc in my lab, together with the Morgan lab at University of Chicago. And I'm going to show you a really simple representation of that kind of signal design process. So first of all, we're going to think this is the simplest cost function that we could think of. So we started there. We have 2 elements. One is a measure of the energetic cost. So if we have, for example, 12 bits that are on, that would be the energetic cost, or 8 for this other example. We have a second element here that is a measure of pattern similarity. So here we again define it very simply by the largest sequence that is overlapping between the 2 species, and actually between every pair of species of fireflies. And because we know the fireflies are broadcasting these signals not necessarily in a synchronized way, we're also looking at all the shifts of the relative starting point between the first and the second pattern. And then we use a well-known approach to generate signals. It's kind of like a Monte Carlo approach. It's called the naming game. And this is how we implemented it for the fireflies. So we have, in this example, 3 species. Each one has an initial random flash pattern. Then we're going to mutate one of these bits to be different. And then we recalculate the cost function and then accept with a certain probability if it actually helped us decrease the cost function. And we repeat this until the system converges or until the cost function reaches a plateau value. So here's an example of such a simulation. Here we have 6 species of fireflies. And at some point, the system kind of snaps into a collection of sequences that are actually kind of periodic. So we were quite surprised that we got this, even though we didn't explicitly require the patterns to be periodic. And we can play this theoretical game of asking questions like how would the number of species or the ratio of the 2 elements of the cost function would affect the resulting sequences, and recently presented this at the Intelligence, Robots, and Systems conference. But we can also use that to sort of reverse engineer the ecological constraints that Firefly actually experienced when they evolved. So here in this very preliminary example, we have 6 fireflies. We know their phylogenetic tree, so we know how the speciation occurred. We know what is their flash pattern. And the simulation and the signal is simple enough that we can actually brute force calculate all the different combinations of the similarity coefficient and the energy coefficient. And then we can compare the resulting sequences from the simulation to what fireflies actually do. And wherever we have a peak here, that would be an indication that there was perhaps these kind of evolutionary constraints in terms of the similarity coefficient and the energy coefficient. All right, so that's it. I showed you a couple of examples of work in my lab, several from chemical communication, where we saw that the signal directionality can be a means to increase robustness when we are dealing with the localization of the target. Also, aggregation can be a means to decrease the convergence times when we think about distribution networks in bees. And then with fireflies, visual communications, we saw a recording of synchronous fireflies where we identified the emergent signal periodicity in collective rhythmic communication of fireflies. And lastly, I showed you a Firefly-inspired vocabulary generator for communication in multi-agent systems. So I hope that with all of these different examples, I was able to convey that these are interesting systems to study, not just because we can learn something about insect communication or animal communication, but perhaps we can also leverage that understanding towards bio-inspired designs and swarm robotics and distributed communication and so on. With that, I just want to acknowledge all the wonderful postdocs and graduate students I have in my lab. This is definitely a collaborative effort. And we also have a bunch of beekeepers and undergrad students and even high schoolers that are just excellent. And I'm feeling very fortunate to have them in the lab. I want to thank my collaborators, sources of funding, and that's it. Thank you for your attention. I'm happy to take any questions now.
All right. So we have some time for questions. I wonder if you might unshare your slides so we can go into gallery mode and see each other. All right. And if people would like to raise their hands, I can call on people. Oh, come on now.
Tom has a question.
Oh, okay, Tom.
Orit, first of all, spectacular combination of everything from computation to principles from physics to ecology and evolution and some really high-tech imaging. I was pretty much blown away by it. But I did have a question, and because in the introduction you said that you would tell us about signal to noise ratio, and I didn't hear much about that, and I wondered, just from, zooming out, not getting into the specifics, but I can imagine that evolution probably optimizes signal to noise only to the extent that it makes a difference, because it costs more, right? You pay a higher price to make it better than you need to. So for these different systems, is there any similarity? Do you need a tenfold signal to noise to get these communication systems to work robustly, or is it all over the map?
Yeah, that's a really good question, and I didn't quite get into that because we don't know. So we're kind of using the signal to noise problem and wanting to maximize it as a guiding element or concept. And for the Fireflies, for example, if you can imagine that you have a certain number of channels that want to communicate a particular message, then if all of them are producing the same message and synchronously, then the output is just going to be the message times the amplitude that is defined by the number of channels you have. So that's one really simple way to increase the signal to noise in a very hand-wavy way. And similarly with the non-synchronous Fireflies, now we're starting to get into a situation where we have more than one species perhaps coexisting in the same field. So how do you increase the signal to noise ratio in the presence of these other species is kind of the question, but we don't know the answer yet. I think it's just the start.
Thank you.
Time for a couple more.
I have a question.
Go for it.
Really great talk, thanks, Orit. And I mean, it seems to me that you have shed some new light on this Firefly synchronization problem, which has been studied for a long time. And like this wave propagation, I'm very interested in whether or not, well, first of all, like you've narrowed down that there's a frequency of oscillation that sort of is recruited by more agents as it's sort of moving across this spatial front. And have you done any analysis into whether or not the Fireflies could be building like their own model of like anticipating when this sort of blinking is going to start, or whether this is just, I mean, maybe that would be a latency question and not really a front movement question, like on the global scale, if this makes sense. Does that make sense?
I think so, yeah. So we're definitely interested in that question, but we don't know the answer yet. If we look at what I mostly showed you was what happens between bursts, but actually, if you look just within one burst, Fireflies are really good at synchronizing their flashes, even if there's a small number of Fireflies in the system, even if there's 5. You can see that the flashes just within the burst are really on, they're on the same rhythm. But what seems to be less rhythmic is the time between the onset of one burst to the next one. And actually, I think Owen is here. Owen is currently working on testing exactly, getting data that would help us answer that question by using artificial light. We use LEDs, which the Fireflies respond to, thankfully. So we can really design the signal that they're going to experience, and by their response, maybe we can answer that question. But again, this is kind of preliminary. So I don't know the answer.
They might be building their own model of this state of the this sort of oscillation on the per pulse level, but not on the sort of broader when do the pulses begin in this big. Okay, that's really cool. Yeah, thanks.
Yeah, exactly.
Eva?
Hi, that was really amazing, Orit. I really love this information. And I have a question. So one of the things that you mentioned when you were talking about the Firefly evolution was factors that might differ between males and females in across or ecological reasons for the speciation and things like that. And so my question is, my very long remembering, trying to remember what I used to know about fireflies, are you able to address sexual selection in these models where there's a role of female preference for flash patterns, and what is known in the model of speciation is there female choice driving some of the synchronizations, or and do you have models that can include that as well?
Yeah, so that's another really interesting, I think, extension of what we've been doing so far. Because I think so far I really just told you about males. Females would have their own pattern that they produce, and it's also species specific. What is known from the literature is that females prefer punctual, males who produce a punctual flash pattern for that specific species. So there's definitely some selection happening on whatever pattern that the males are producing, but there hasn't been really, there hasn't been any very long-term analysis and experiments of the actual reproductive success of the swarm as a whole, which I think is another really fascinating question. Yeah, so I just want to mention that in terms, again, with LEDs, we're doing some pilot experiments now where we can simulate the pattern of a female firefly with the LED, and we can see how the males respond. So again, maybe that's kind of a way to start asking those questions.
Great. Well, we're about out of time, so let's thank Orit again and go out and flash.
Thank you all very much.
Great job, Orit.
Yeah, that was brilliant, Orit.